Hello students, welcome to EPG Pathshala. I am Dr. Radhika Bakshi from University of Delhi. Today we are going to discuss about the structure and role of the mitochondrion from the paper Thermodynamics of Living Systems and Bioenergetics. So students, let us see what we are going to learn in this module. First, the detailed structure of the mitochondrion. Then, the mitochondrial genome. Then we will go on to study the main functions of the mitochondrion, which is ATP synthesis. There are also some tissue-specific functions of the mitochondrion. Example, thermogenesis, synthesis of steroid hormones, and apoptosis. Lastly, we will study about the origin of the mitochondrion, which is given by the endosymbiotic theory. The energy to convert ADP to ATP is produced by two main processes. One, aerobic oxidation, which occurs in nearly all cells, and photosynthesis, which occurs in the leaf cells of plants and certain single-celled organisms. Glucose is the primary fuel, and its initial steps of oxidation takes place in the cytosol of the cell by glycolysis in both eukaryotes and prokaryotes without the requirement of oxygen. The final steps of oxidation requiring oxygen generate most of the energy and take place in an organelle present only in eukaryotic cells and not in prokaryotic cells, which is called the mitochondrion. These are among the larger organelles in the cell. Each mitochondrion is roughly the size of an E. coli bacterial cell. Many mitochondria are present in most eukaryotic cells, occupying up to 25% of the total volume of the cytoplasm. Various three-dimensional images obtained by different imaging techniques have revealed a greater variation in mitochondrial size and shape. A number of variously shaped mitochondria are seen clustered around the nucleus when living cells are stained with mitochondrion-specific fluorescent dyes. The tissues having a high demand for aerobic metabolism, example, the brain, the eye, heart, and the skeletal muscle, contain many hundreds or thousands of mitochondria in each cell. Also, the cristae of these mitochondria, which are the enfoldings of the inner mitochondrial membrane, are more densely packed. In these tissues having less active metabolism, example the skin, there are fewer mitochondria, each with fewer cristae. The mitochondria usually divides by fission, like the bacteria, during cell growth and division. Under certain conditions, individual mitochondria also fuse to form larger, more extended structures. The structure of mitochondria. Mitochondria are large enough to be seen under a light microscope, but detailed structure can be viewed only with an electron microscope. High-resolution electron micrographs suggest that the mitochondrion contains two membranes, the outer and the inner membranes. The nucleus and the chloroplast also possess double membranes, but majority of the cell organelles like the ER, Golgi, lysosomes and peroxisomes are single membrane structures. Outer membrane. The outer membrane of the mitochondria is smooth and defines the external boundary of the organelle. Small molecules like sugars, salts, nucleotides, and coenzymes are able to penetrate through the outer membrane because of the presence of small pores called porins. These porins are transmembrane channel proteins which readily permit the passage of ions and most small hydrophilic molecules up to a size of 5,000 Daltons. Similar proteins are also present in the outer membrane of the gram-negative bacteria and in the outer chloroplast membrane. The presence of porins in both outer mitochondrial membrane and the outer membrane of gram-negative bacteria supports the view that the two membranes have a common evolutionary origin and supports the theories claiming that mitochondria evolved from symbiotic bacteria hundreds of millions of years ago. This permeability of the outer membrane allows inner compartment more direct access to the substances in the cytosol, and hence the solute composition of the intermembrane space resembles that of the cytosol. The inner membrane. The composition of the inner membrane. Almost 76% of the total weight of the inner membrane is contributed by proteins, a much higher fraction than in any other cellular membrane. The presence of the lipid diphosphatidylglycerol or cardiolipin, which is present in a high concentration in the inner membrane, sufficiently reduces the membrane's permeability to protons. This allows the establishment of a proton motive force across the inner membrane. The inner membrane also has the cristae. The cristae is the infolding of the inner membrane, that is the invaginations of the inner membrane. The area of the inner membrane, including the cristae, is about 5 times that of the outer membrane in a typical liver cell. These cristae greatly expand the surface area of the inner membrane, thus enhancing its ability to generate ATP. Cells of tissues having a greater demand for ATP, like the heart and the skeletal muscles, contain 3 times as many cristae as found in a typical liver mitochondria. The various proteins of the cristae are indicated by the freeze fracture studies. The mitochondrial cristae contain many protein-rich intramembranous particles. Some of these are the F0 F1 complexes or the ATP synthase, which synthesizes ATP, and also the components of the ETC or the electron transport chain, which functions to transport electrons to oxygen from NADH or other electron carriers. The inner membrane is permeable to most small molecules and ions, including protons. The only species that cross this membrane are the ones having specific transporters to move through. Therefore, The major permeability barrier between the cytosol and the mitochondrial matrix is the inner membrane. Some of the impermeable molecules such as ADP and inorganic phosphate pass across the cytosol to the matrix, and molecules like ATP move from the matrix into the cytosol. These move across with the help of various transport proteins located in the inner membrane of the and the cristae. The two mitochondrial membranes, the outer and the inner, define the two submitochondrial compartments, the intermembrane space and the matrix. The intermembrane space. This is the compartment between the outer and the inner membrane. The permeability of the outer membrane allows the inner compartment more direct access to the substances in the cytosol, and hence solute composition of the intermembrane space resembles that of the cytosol. The matrix. This is enclosed by the inner membrane and is the second submitochondrial compartment. It contains the mitochondrial genome and all the machinery for DNA replication, transcription, and protein synthesis, including the ribosomes. It is also the site for the fuel oxidation pathways like citric acid cycle, fatty acid beta oxidation, and amino acid oxidation. It also houses the pyruvate dehydrogenase complex for the conversion of pyruvate to acetyl-CoA. The selectively permeable inner membrane segregates the intermediates and enzymes of cytosolic metabolic pathways from those of metabolic processes occurring in the matrix. The mitochondrial genome. Apart from the nuclear genome, the cell also has genes housed in the mitochondria. The mitochondrial genome is a circular double-stranded DNA molecule. Around 5 copies of this genome are present in each of the hundreds or thousands of mitochondria in a typical cell. Mitochondrial DNA lacks histones and hence is not packaged into nucleosomes. The human mitochondrial chromosome contains 37 genes in all and is 16.5 kb or kilobases long. Various subunits of proteins of the respiratory chain are encoded by 13 of these genes. The remaining genes code for ribosomal RNA and transfer RNA molecules. These are used for protein synthesis in the mitochondrial matrix. Majority of the mitochondrial proteins, around 1100 different types, are encoded by the nuclear genes, which are synthesized on the cytoplasmic ribosomes. These proteins are then imported and assembled in the mitochondria. The mitochondrion has its own DNA and has the machinery for replication, transcription, and translation. Therefore, it can be considered as an autonomous cell organelle. But the mitochondria is not capable of existing independently since most of its proteins are coded by the nuclear genome and are synthesized in the cytosol. Hence, it is not fully autonomous and therefore is called as a semi-autonomous organelle. As we can see in figure 2, the mitochondria outer membrane is freely permeable to small molecules and ions. The inner membrane is impermeable to most small molecules and ions, including protons. The inner membrane contains respiratory electron carriers, complexes 1, 2, 3, and 4. Also, it contains the ADP-ATP translocase, the ATP synthase, and other membrane transporters. The matrix contains the pyruvate dehydrogenase complex, the citric acid enzymes, the fatty acid oxidation enzymes, and the amino acid oxidation enzymes. The matrix also contains DNA, ribosomes, and many other enzymes. It also has many soluble metabolic intermediates. Maternal inheritance. In most organisms, including humans, the paternal mitochondrial DNA is not transmitted to the offspring. This pattern of mitochondrial DNA inheritance is called as maternal inheritance. The reason being is that the mother's egg cell donates the majority of cytoplasm to the embryo. The mitochondria contributed from the father's sperm are usually destroyed for unknown reasons. The various functions of the mitochondria. The metabolic role of the mitochondria is very critical. This is reflected by the fact that defects in the mitochondrial function have serious medical consequences. They are central to neuronal and muscular function and also to the whole regulation of the whole body energy metabolism. They are central to the neuronal and muscular function and also to the regulation of whole body energy metabolism and weight. Compromised mitochondrial function has been implicated in human neurodegenerative diseases, cancer, diabetes, and obesity. One of the theories of aging is also based on the gradual loss of mitochondrial integrity. The various roles of mitochondria are the most important being the production of ATP. ATP is synthesized in the mitochondria through the process of oxidative phosphorylation. All the oxidative steps in the degradation of carbohydrates, fats, and amino acids converge at this final stage of cellular respiration. ATP synthesis is driven by the energy harnessed from the oxidation of various fuel molecules. The figure 3 depicts the various stages of metabolism. Stages 2 and 3 occur in the mitochondria. All the pathways of fuel oxidation, as seen in figure 2, except glycolysis, which take place in the cytosol, occur in the mitochondria. Hence, mitochondria plays a central role in the oxidation of fuels and generation of ATP. The process of ATP synthesis is based on the chemiosmotic theory, which was postulated by Peter Mitchell in 1961. According to this theory, the transmembrane differences in proton concentration is the reservoir for the energy extracted from biological oxidation reactions. The various stages of oxidative phosphorylation are: the first one, flow of electrons through a chain of membrane-bound carriers. This is called as the electron transport chain. The second, the downhill or exergonic electron flow makes free energy available for the uphill transport of protons across the proton impermeable inner membrane. This conserves free energy of fuel oxidation as a transmembrane electrochemical potential. Specific protein channels allow the transmembrane flow of protons back down their concentration gradient in the third part. This provides the free energy for the synthesis of ATP, which is catalyzed by a membrane protein complex called the ATP synthase. This complex couples proton flow to phosphorylation of ADP. The complexes of the electron transport chain. Gentle treatment of the inner mitochondrial membrane with detergent resolves the 4 unique electron carrier complexes. Each of these protein complexes is capable of catalyzing the transfer of electrons through a portion of the chain. The various complexes of the electron transport chain. The first complex or complex 1 is called as the NADH dehydrogenase. It is a large enzyme composed of 42 different polypeptide chains and catalyzes the electron transfer from NADH to ubiquinone. Ubiquinone is a lipid-soluble electron carrier and is also called as coenzyme Q. It also pumps 4 protons from the matrix to the intermembrane space. The matrix thus becomes negatively charged and the intermembrane space acquires a positive charge. The complex 2 of the electron transport chain. It is called as a succinate dehydrogenase complex. It is the only membrane-bound enzyme of the citric acid cycle, all the other enzymes being soluble in the matrix, and this complex is also part of the electron transport chain. Thus, it has a dual role. It catalyzes the conversion of succinate to fumarate. The electrons are transferred from succinate to ubiquinone. Complex 3 of the electron transport chain. This is called as the cytochrome BC1 complex. This complex couples the transfer of electrons from reduced ubiquinone, which is QH2, to another electron carrier, which is cytochrome C. Cytochromes are proteins having a characteristic strong absorption of visible light due to their iron-containing heme prosthetic groups. Cytochrome C is a soluble protein of the intermembrane space and associates through electrostatic interactions with the outer surface of the inner membrane. The cytochromes of type A and B are integral proteins of the mitochondrial inner membrane. Complex 3 also pumps 4 protons from the matrix to the intermembrane space. Complex 4 of electron transport chain, or the cytochrome oxidase. In the final step of respiratory chain, complex 4 carries electrons from cytochrome C to molecular oxygen, reducing it to water. Molecular oxygen, therefore, acts as the ultimate acceptor of electrons. Complex 4 also pumps 2 protons from the matrix to the intermembrane space. The energy of transfer of electrons is efficiently conserved in a proton gradient. For each pair of electrons transferred to oxygen, 4 protons are pumped out by complex 1, 4 protons are pumped by complex 3, and 2 protons are pumped by complex 4. This type of electrochemical gradient generated across the inner mitochondrial membrane constitutes the proton motive force. This proton motive force has 2 components. First, the chemical gradient, which is due to the difference in concentration of the chemical species, the protons, across the membrane. The second being the electrical gradient. It is due to the separation of charge across the membrane created when a proton moves across the membrane without a counter ion. Therefore, the matrix becomes negatively charged and the intermembrane space becomes positively charged. The synthesis of ATP. The chemiosmotic model proposed by Peter Mitchell explains the mechanism of the coupling of proton flux to phosphorylation. According to the model, the electrochemical energy inherent in the difference in proton concentration and the separation of charge across the inner mitochondrial membrane, that is, the proton motive force, drives the synthesis of ATP as protons flow passively back into the matrix through a proton pore in the protein complex, which is the ATP synthase. Both ATP synthesis and electron flow through the respiratory chain are coupled. Neither of these 2 processes can proceed without the other. The complex 5 or ATP synthase. The mitochondrial ATP synthase is an F-type ATPase and is present as a large enzyme complex of the inner mitochondrial membrane. It catalyzes the formation of ATP from ADP and inorganic phosphate and is driven by the flow of protons from the P phase, that is facing the intermembrane space, to the N side, which is facing the mitochondrial matrix of the inner membrane. It has two distinct components, F1, which is a peripheral protein, and F0. The naught or O denotes the oligosensitivity. O denotes oligomycin sensitivity. So, the F0 or FO spans the membrane. The tissue-specific functions of mitochondria. Although ATP production remains the central role for the mitochondria, it has some tissue-specific functions as well. First one being thermogenesis. In the adipose tissue, the mitochondrion generates heat to protect vital organs from low ambient temperatures. Most newborn mammals, including humans, small vertebrates, and hibernating animals, possess a brown adipose tissue or BAT, which constitutes a significant proportion of the adipose tissue. It can be distinguished from the white adipose tissue by its smaller diameter and polygonal adipocytes. These also store triacylglycerol, but in several lipid droplets per cell instead of a single central droplet as in the white adipose tissue. BAT cells also have more mitochondria and a richer supply of capillaries than the white adipose tissue. The characteristic brown color of BAT is due to the cytochromes of the mitochondria and the hemoglobin in the capillaries. The brown adipocytes uniquely express the gene USP1, which encodes the mitochondrial uncoupling protein called thermogenin. This protein is responsible for thermogenesis, which is the biological generation of heat by uncoupled oxidative phosphorylation. In white adipose tissue, the protons pumped out of the mitochondrial during electron transfer re-enter the matrix through ATP synthase protein, and hence energy of the electron transfer is conserved in the synthesis of ATP. In contrast, the thermogenin in brown adipose tissue provides an alternative route for protons to re-enter the matrix and bypasses the ATP synthase. The energy of proton gradient is thus dissipated as heat, which maintains the optimal body temperature when the ambient temperature is relatively low. At the time of birth, the brown adipose tissue or BAT represents 1 to 5% of the total body mass. It is located in regions where the heat generated by thermogenesis ensures protection of vital tissues from lower ambient temperature when the baby is born. Young adult humans have less BAT deposits from 3% of all adipose tissue in males to 7% in females, making up less than 0.1% of the body mass. Interestingly, BAT in adults can apparently be activated by cold exposure and suppressed by increasing the core body temperature. Another function of mitochondria, which is tissue specific, is the steroid hormone synthesis. In tissues like adrenal glands and gonads, mitochondria are the sites of steroid hormone synthesis, including steroid hormones, glucocorticoids, mineralocorticoids, and vitamin D. Cholesterol, or a related sterol, is the precursor of all these compounds in a series of hydroxylations catalyzed by enzymes of the cytochrome P450 family. All these enzymes have a critical heme group and absorb light maximally at 450 nanometers. Hence the name P450. There are many P450 enzymes, all situated in the inner mitochondrial membrane, with their catalytic site exposed to the matrix. Cells synthesizing steroids, which are also called as steroidogenic cells, are rich in mitochondria, which are generally larger in size than those in other tissues. These also have more extensive and highly convoluted inner membranes. Another tissue-specific function of mitochondria is apoptosis. In most or all tissues, they are key participants in apoptosis. Apoptosis, also called as programmed cell death, is a process in which individual cells die for the benefit of the organism. An example being in the normal development of an embryo. It may be triggered by an external signal acting on a plasma membrane receptor, or by internal events like DNA damage, viral infection, oxidative stress from accumulation of reactive oxygen species, or other stress such as heat shock. Role of mitochondria in apoptosis. When a stressor signals for cell death, there is an increase in the permeability of outer mitochondrial membrane. This leads to the escape of cytochrome C, which is one of the electron carriers of the electron transport chain, from the intermembrane space into the cytosol. This acts as a trigger for apoptosis by stimulating the activation of a family of proteases called caspases. Origin of mitochondria. Altman in 1890 referred to mitochondria as a primitive organisms analogous to bacteria. He suggested that mitochondria can exist as free-living forms outside the host cytoplasm. In early 1960s, the discovery of the mitochondrial DNA and the similarities between the protein and nucleic acid synthesis between mitochondria, chloroplasts, and bacterial cells prompted scientists to further look into the evolutionary link between the mitochondria, chloroplasts, and bacteria. Some of the biochemical similarities between these three are they have similar to the bacterial DNA, the mitochondrial DNA and the chloroplast DNA is also circular and not complexed with histones, as is the case in eukaryotic nuclear DNA. The base sequence of mitochondrial and chloroplast ribosomal RNAs resemble bacterial ribosomal RNAs more closely than eukaryotic cytoplasmic ribosomal RNAs. Another point, the ribosomes of mitochondria and chloroplasts are usually smaller than the ATS cytoplasmic ribosomes, a property which is similar to that of bacteria. Another point of similarity is bacterial and mitochondrial RNA polymerases are inhibited by the antibiotic rifampicin, but the drug doesn't inhibit the eukaryotic nuclear RNA polymerases. Apart from the above mentioned biochemical similarities, mitochondria and chloroplasts resemble the bacteria in shape and size. Also, the plasma membrane of bacteria and the inner membrane of mitochondria possess protruding spheres containing the ATP synthase involved in oxidative phosphorylation. The same basic mechanism is used by mitochondria and chloroplasts and bacteria for ATP synthesis, that is chemiosmosis, or also called as chemiosmotic coupling. In this mechanism, a proton concentration gradient and an electric potential, that is the voltage gradient, across the membrane collectively termed as the proton motive force drives the synthesis of ATP. The endosymbiotic theory. The similarities between the mitochondria, chloroplasts, and bacteria have given rise to a popular hypothesis that these organelles arose early in evolution of eukaryotic cells by the endocytosis of bacteria, which were capable of oxidative phosphorylation in the case of mitochondria or photosynthesis in case of chloroplasts. This endosymbiotic hypothesis, as it is called, states that the inner mitochondrial membrane would be derived from the bacterial plasma membrane with the globular F1 domain still on its cytosolic face, pointing towards the matrix space of the mitochondria. This can be explained in this figure. So students, let us summarize what we have learned in this module. The final steps of oxidation requiring oxygen generate most of the energy in the cell and take place in an organelle present only in eukaryotic cells called the mitochondrion. It is a double membrane, bean-shaped organelle. Its outer membrane allows small molecules to pass freely with the aid of transmembrane proteins called porins. The inner membrane is folded into invaginations called cristae and houses all the components of the electron transport chain. The inner membrane is selectively permeable and has membrane transporters for specific molecules. It is also the main site for ATP synthesis by chemiosmosis. The mitochondrial matrix contains enzymes for the oxidation of acetyl-CoA with the help of Krebs cycle, also lipids and amino acids. The matrix also contains DNA, RNA, ribosomes and enzymes involved in nucleic acid and protein synthesis. The human mitochondrial chromosome contains a total of 37 genes and is 16,569 base pairs long. Various subunits of the proteins of the respiratory chain are encoded by 13 of these genes. ATP synthesis is the main function of the mitochondria. The basic mechanism for ATP synthesis is chemiosmosis or chemiosmotic coupling. In this mechanism, a proton concentration gradient and an electric potential, that is the voltage gradient, across the membrane, collectively termed as the proton motive force, drives the synthesis of ATP. Apart from this main function carried out by all cells, the mitochondria also has some tissue-specific functions. In the adipose tissue, it generates heat to protect vital organs from low ambient temperature. In adrenal glands and gonads, mitochondria are the sites of steroid hormone synthesis, including steroid hormones, glucocorticoids, mineralocorticoids, and vitamin D. In tissues, they are in some tissues, they are also the key participants in apoptosis. Apoptosis, also called as programmed cell death, is a process in which individual cells die for the benefit of the organism. Example, in the normal development of an embryo. There are similarities between the mitochondria, chloroplast and bacteria. This have given rise to a popular hypothesis called the endosymbiotic hypothesis, which states that these organelles arose early in evolution of eukaryotic cells by endocytosis of bacteria capable of oxidative phosphorylation in case of mitochondria or photosynthesis in case of chloroplasts. Thank you.